Good morning, all. I think, can you hear me? Okay. Section because today is the welcome to all this online CSIR coaching life science. This is section 1, day 2. Now we are going to discuss about the cell communication and cell signaling. Uh, if you have any doubts, please ask at the end of the session because in between we can't answer, right? So only I am telling this. Actually I am in PPT window, so I am not able to see your comment. uh, please follow these steps. If you have any interruption, please, I will rectify it. நேற்று நிறைய பேர் கேட்டிருந்தாங்க, வி நீட் இட் இன் தமிழ். இஃப் பிட்வீன் தமிழில் பேச சொல்லி கேட்டிருந்தாங்க. In this online, if anyone, if you not understand the Tamil, please tell in chat because it's a very important one. So only I'm telling this. If is there any people you don't know Tamil? Good morning, all. I think. Can you hear me? Okay, fine. Okay, now we are going to the section cell communication, cell signaling, because yesterday I'm told we are going to see the fourth unit in first day. Okay, first the fourth unit contains the different sections, that is fully cell communication and cell signaling. Okay, cell means that is includes a bacterial cell and also the plant cell. All cells are included in one particular area, so only they are telling the cell communication and cell signaling. Bacteria can interact with human, bacteria can interact with bacteria can interact with in various way. We can tell which is how they are communicated. In this I'm just explaining the example. After finishing this section, I will send the PDF with the particular disease name and the bacteria to your mail ID or the session or the Google Classroom, because many terms are here in cell communication and cell signaling. I'm not cover all the um, I'm not cover all the sessions, so I'm just telling the example in during this 1 hour. After finishing this session, we please refer some I will send some article with the bacterial disease name, bacterial cell surface molecules related details. Okay, I'm just putting this for your refreshment, because fully upcoming slides are fully theory, so I want to explain with you this representation. You all know, right? This nowadays we are mostly fully depends on mobile. This mobile having the facility to மேம். ஆக்சுவலி எஸ்டர்டே சேனல்லே பார்க்கலாம் மேம். இல்லை மேம், இப்போ லைவ்ல தான் இருக்கு. ஓகே மேம், 1 மினிட் மேம். அந்த சேனலில் ரெஃப்ரெஷ் பண்ணிங்கன்னா ஃபஸ்ட்டு அதில் வரும் மேம். I mean reload பண்ணா இல்ல மேம். ஒரு நிமிஷம் மேம், நான் உங்களுக்கு இதில் வாட்ஸ்அப் சென்ட் பண்ணுவேன். ஓகே ஓகே. Actually, sorry friends, some of them not getting the link, so they are asking the link. Please wait few minutes. Sorry. Okay. Now we are start this section. Okay. Everything we need a signal, because if we are if you are not able to connect with the signal, we can't communicate each other. The same principle is happening in the bacteria also. Every bacterial cells, viral cells, and also many human cells, plant cells, all cells are finding the signaling molecules. These signaling molecules interact with the passing the signals, because if the bacteria in the antigen like structure, means that chemical structure that easily interact with human cell. For example, now in a cell interaction means that virus and human cell also interact with each other. I'm going to explain with this small example, which is in current situation. We are all know, right, the coronavirus. This coronavirus is having cell surface signaling molecule that's first interact with human cell surface. The animal cell into human cell surface molecule, I mean that virus cell molecule interact with human cell surface. This particular protein, that means the spike protein, that pass the signals to our human cell surface protein, that is ACE2 receptor, that makes the interaction over for further infection. So every cells needs a signal. So only I am telling this image. Okay. Like our scientists are reported, like our human generation, the bacteria is also having many generations. Okay, first generation, second generation. The first generation bacteria are not able to communicate with all human, but are nowadays based on the demand, their bacteria are regenerated, that now current generation of bacteria are interact with human easily. here that I'm telling this because this image represent the first in the just refreshment. Next upcoming slide I'm going to tell the fully theory part. Can you able to see me or hear me? Sorry for the interruption. Okay. Yeah. Okay, fine. Now we are going enter into the session. Sorry for sorry for the disturbance. Okay. In Unit 4, they are having the 5 different area. First one is host interaction, the interaction of cell with bacterial, viral, and any disease related topic. The green topic highlighted thing are the say the infection related disease related topic. In that part we are going to discuss how to the bacteria interact with animal, how to the bacteria interact with the plant, how the cell bacterial are going to interact each other. Then the next part is going to the blue highlighted but is a fully cell signaling hormones. It's are related to fully to human cell surface related topic. The blue color highlighted is the next tomorrow part. Today we are going to discuss about the parasite interaction recognition. Okay. Next, these are the upcoming topics because some of them are asking please tell the next day topic, so only I am subjected. And separately I'm telling this because the first the green highlighted is the first topic of 4th unit, highlighted is the second topic of the next topic of unit unit 4. This cellular communication and regulations of hematopoiesis are the third part. Another is a cancer related part is a fourth one. The fifth one is a very important topic, that is the immunology related topic. We have a separate day. And now I'm entering into the fourth unit, the first session. The fourth unit, first who is studied about the first question, who is studied about the host pathogen interaction. He is you all know the coach pass slate, right? Many of the students are no, staffs are no, because this the coach pass slate first studied the microbial interaction. Okay, he explained why I am telling this in this PPT mean. First we want to understand how what is the mechanism behind the host pathogen interaction. Okay, what are the needs behind the interaction. Because if the microbe want if in he tell the coach, as per the coach pastillate report, many of the diseases are caused by the microbial system. And he reported every case is having the microbes in their particular. If you got a sugar infection, sugar wound, if wound healing property, or maybe some of them know, if you having any wound means in that particular area many bacteria are present in particular portion. So it's a for one example. Another thing is if you have any intestinal problem means in that also some of the microbes are responsible. It's a very one important thing, because all of the diseases are having the some responsible related to the microbe. The first is the microbe must present in case of diseases. Next thing, if the microbe want to grow means we need a pure culture. If pure culture means, why I am telling this, we are all you may know about the bacterial fighting, right? If அதில் ஒரு பாக்டீரியா இருக்குது அப்படின்னா அந்த another bacteria இருந்துச்சுன்னா அதை க்ரோ பண்ண விடாது. ஸோ தட் இஸ் கால்ட் அஸ் so only they are needing the pure culture. Okay. Some diseases must be produced when the pure culture of organisms is in the sustainability host. Okay, எல்லா இடத்துலையும் வளர்ந்துராது. ஒரு பாக்டீரியா வேணும், ஒரு பாக்டீரியா want to grow அப்படின்னா that need a specific host. Okay, corona, I am going to corona. Corona can't be able to grow in any plant, that can't grow in any some other animals. Now till date no one is reported in human. There is and bacteria and that bat also not affected by coronavirus. It's just a host that they are mutually understandable that coronavirus not affecting the bat, but coronavirus affecting the human. So we are enemies, they are friends. That kind of interaction that happen in coronavirus. Then another one, so that every organism need a specific growth. Then organism must be recovered from the after the growth in particular host. We can easily recover from particular host. Easily recover means, okay, next I'm going to with example. Because intestine having the Lactobacillus species that is recovered from cow through the milk. That milk give us our digestion system. So it's are correlated and interlinked in with particular cycle. Okay, next in this course postulate they are explained some experimental values, because all of them related to experiment, right? CSIR in CSIR examination point of view, they are asking their statement, okay, this microbe is present in particular animal, that animal having this disease. Okay, how to correlate that disease is highly occur in particular animal. What is the remedy? What is the output? It is understandable, or they are not understand, and they are not acceptable host. What is these kind of questions they are asking. I'm entering into the topic because the whole first we are understand the host pathogen interaction. First, every disease is having the microbial presence. Then if you want to grow the microbe means you need a pure culture or any host cell. If any host cell can support microbial growth means that host will affect or interact the microbe. One is against one, it is another is the interaction one. The host and host and microbe are having the friendship that interact each other. The host and microbe having the opposite taste, that is affect. That microbe may be affect the host, or host may affect the microbe. These are the two possibility. Then next is the host. So only they are telling they need a specific host. Okay, next we are moved to the components of host pathogen interaction. First is invasion. Then second is evasion, microbial replication, then host immunology. Okay, these four are interlinked. Because invasion means attachment of microbe on the cell surface of the host surface. Then after that that local tissues that spread into the local tissue. After that adaptation, that is replicate in the particular host cell. After that it will after the replication of microbe, that microbe will enter into the human when in the whole body. So the whole body get disturbed, so immune system will activate it. It's a basic steps. Okay, now we enter into the another part of the unit 4. It's a very in the topic la irundhu Definitely they are asking the question, because last I referred last 3 years question all the questions they are asking this kind of questions, because the mutualism, commensalism and parasitism, because they are asking, they are given the name. Okay, this interact, okay, cow and bacteria interaction, another side fusion. It's an it's a match the following related question. All the three questions they are asked, the main related format. One side they are given the cow and bacterial interaction, another side they are mentioned the bacterial name. That is one kind of question. Another question is, one side they mention cow and bacteria, it's a mutualism or commensalism, or you need to find out. This kind of questions are there. Next is during the mutualism, both the microbes and the body will work together. Okay, that is, there is no negativity, fully they are interact, that they are friendly. So both the microbe and the host may be plant or animal. That is the thing. After interaction, they are not affected. The host also benefited, the microbe also benefited. For example, in the cow and bacteria, in a rumen of cow, there are many bacteria are there. Specifically, Lactobacillus species are the most important one. In this Lactobacillus species, having the cellulose facilitated digestion. Cellulose facilitated digestion, why we needed in the particular cow. Okay, the cow intestine, a cow major food of the cow is grasses, fiber related or carbohydrate rich components, because that is we all know the all kind of foods are carbohydrate. This this cow specifically eaten the specifically take the particular carbohydrate rich food. So that need, what is the role of lactobacillus? That gives the facilitated digestion in particular cow. It's a major thing. In this particular para, 3 type of questions are, I'm just giving the example alone. It's also the one question they asked the role of lactobacillus in cow. So only I am telling this. Well, our eye flora having the flora have habitated in the eye, but the eye not get any infection. Already usually the flora is present in our eye, that flora get benefited, but our human host will not get benefited. And also it not affected by interaction. Host will not get affected by interaction. Some of them may be asking in the commensalism whether the host will get interaction or get affect or not. Okay, this kind of questions are there. In this also they mention the flora, specifically mention the flora name. In upcoming slide, what kind of floras are there, and how can we explain it, how can we explain it. I'm just telling that. I will tell in next slides. Okay, next is the parasitism. Parasitism is one organism another organism will affect. Okay, now coming to the again corona. Okay, during the corona, our human system will affected. the coronaviral system will get benefited. That is the because here mutualism, commensalism, and the parasitism. These three are the major repeatedly asked question. You can see anywhere in any question, December question or June question. In all the questions they are asked the about particular infection. So this kind of questions and infection rate in the mutualism. You can in mutualism that not affect the host cell, right? So the microbial presence is high. In mutualism, that microbe will not affect the host, so the count of the microbe is high. Another thing, commensalism. In this commensalism, it's not favorable because it's not it's not a regular one. The host will get a benefit or the microbe will get benefit, so that is not shown in all cases. Another one is the one microorganism will benefit. It's in this case the microorganism count is very less. The presence of microorganism count is very less. For example, in this corona infection, if the only one viral attached on the host cell, that will replicated after 14 days. So the presence of count is very low. The interaction count is very low, but the infection rate is high. These are some technical questions. Oh, sorry. Now please check it now whether now it is okay. So now only I am seeing your voice. Please check it now. Now I am check my voice, check my internet connection. Please tell whether I am can you hear my voice? Okay. Okay, I'm go with okay. Thank you so much. Sorry, actually, here due to the heavy rain, we are get some internet connection problems only. Sorry. Thank you so much. Your reply. Now I am move to the next slide. Okay, these are the normal flora. In normal flora, they are having two types. If these are fully interlinked with the cell host pathogen host and microbe interaction. It's a flora present in our eye and also present in many animals. So I'm telling this, the normal flora having the resident two types. That one is a resident flora, another one is transient flora. Flora is survey one particular extent period it will survey. Thank you for your response all. The resident flora survey for extended period. The transient flora only for a temporary period. Apart from the temporary and extended period, we want to understand resident means at a full once it's enter into our human body, that fully survey or the whole throughout the or her particular life. Okay, for example, in our intestinal we are having the E. coli, for a digestion in the digestion system, and also we have n number of microbes in our intestine, but that are not affect the human cell, but that are present the whole life. So that kind of is a residential flora. The another one is a transient flora. For example, through one example for this is, if you are taken palm sap, okay, palm sap having the different microbes, that bacillus kind of species, bacillus species, the palm sap having the bacillus species, that is not digested by our gut system, our intestinal system. That palm sap enter into the human body, that will survive particular period, that but not affect the human, that and also that will that have get the glucose and polysaccharide to digest digest our system and digest system. The residential flora is already present in our intestinal, it's present throughout the year. Transient flora, if okay, lactobacillus, if you take a curd, means that enter into the intestine, that microbe will get benefit, and also human also benefited. Sometime that the one case is no, the microbe will not maybe not benefited, right? That only utilize the glucose or carbohydrate from our human body, and it will multiply in intestinal system, then automatically excreted. So these are the transient flora. Next one is host defense system. After once, if any microbe enter into our human body or any plant and animal system, that host system will activate it. Defense system. The host defense system means we are having immunity, we are having the cell surface signaling, that signaling making the response. That response is expressed in different way. These expressions are classified into 4 types: skin, the mucosal secretion. Skin or mucosal secretion means, okay, many of them having the allergy due to the some foods. Sometimes if you are taken a fish, you get allergy. If you are taken any some of them having allergy due to the brinjal. So these are the host defense system expression. This host defense system expression happen. How it happen? Okay, once your digestive system accept the particular allergic food, our system get signal, okay, I got some unwanted food. That signal enter to your brain system. That is the major main part, right? First, the allergic food enter into your stomach system. Your stomach system have the passing the signal to your brain. Your brain, okay, we are some unwanted thing enter into human body. So you that pass the signal to macrophage cells. That macrophage cells are make a inflammation. This inflammation cause the allergic on your skin. Is one kind of thing. Okay, another kind of thing is a mucosal secretion. Okay, if you this is related to common cold and cough. Okay, this if any common cold and cough causing microbe enter into our nose or throat, that causing the that stimulate our human immune system. This human immune system present in gut and nose, they are secreting mucosal to stop the microbial spread into our system. Okay, we are for that mucosal system having a sticky and sticky in nature, that is make a barrier. Through that barrier, the microbe can't enter to human body. that are the upper defense system. The mucus are present in our gut and the nose, stop blocking the entry of microbe into human system. That is one. Another one is non-specific local response. Non-specific local response, the one example is okay, the pH variation. pH variation means our digestion system having the two pH 3 to 4, right? If you are taken abnormal microbial contained food, that make a food poison, right? That during the food poison, the pH variation happen in our intestine. Another one is the non-specific inflammatory response, and the inflammatory response is also our itching related problem. Then specific immune response is a very important one. We will I am giving an example for this at the end of the 4th unit. That caused by the lymphocytes related system. Okay, pathogen defense system having the pathogen also having some virulence factor, that is promote the disease formation. Disease formation means if I'm related to current experiment, if the corona attached to the human cell, that not causing the disease. So if that want to promote the disease now, that want particular signals, that want particular foods to replicate, that microbe need particular enzymes for their replication. That is called as the pathogen defense, the virulent factors. Ability to pathogen to adhere on the host. Okay, the bacterial cell surface want a surface protein. Surface protein, that is in current scenario, if bacterial we all know interaction of bacterial attachment on the cell surface, or bacteria will attach on the cell surface with the flagellus or particular lipid membrane, the membrane receptors. In coronavirus, spike is the attachment protein, the adhesion protein. The ability of pathogen to colonize. Colonize means it want to improve their count. It want to replicate in many count. These are all the particular thing will happen in particular host pathogen colonization. Colonization need enzyme. The question they are asking the question related to that, because many different organisms needs different enzyme for their colonization. That kind of questions will have appear in particular CSIR examination. The ability of pathogen to evade the host system. Evade the host system means that want to adapt and improve the count, and that after the replication that want to evade from the host cell, because that for a long time that is not able to present in particular host. One thing that want to digest the and blast the host cell, or it may be lytic cycle or lysogenic cycle. Okay, next is a oral plan. First, transmission. Okay, it want to transmit. It may be transmitted through water, it may be transferred through the air. Every disease, this kind of questions are there present, because it depends on this related to this transmission related question. They are asking, but they are given a particular disease name or microbe name. They are asking the mode of transmission. It's a related question. Also send the question to you in through the particular Google Classroom or mail, because transmission is caused by many ways. That may be attachment, direct attachment, that may be caused by water, that may be a soil, that may be the transmission route, it may be a air. Okay, these are the transmission medium. What is the transmission route? Okay, it may transmit through your eye, it may transmit through your gut, it may transmit through your nostril. Okay, many routes are there. Questions எப்படி இருப்பாங்க அப்படின்னா, the transmission, they are given a transmission route. Okay, here this also the match the following like question. They given one particular disease name and transmission route. Likewise, 4 different diseases, 4 different route of transmissions. We just want to match the following the particular disease route of transmission. Another one is adhesion and invasion, colonization, evasion, and host defense system, and damage and disease to the host tissues, exit from the body, then survey in the environment. These are the step by step process. Okay, why you tell this? You can ask, right? Why I want to learn this particular steps? They are asking another way. They are misarranging this, given in 4 options. One kind of question is asked in the last 2 year back. They are asked in one particular question. They are rearranging these steps and asked given in option. You need to find out the particular correct answer. It's a task, so only we need it. First, transmission. First, the microbe want to spread. Then adhesion, the microbe want to attach on the cell surface. Then invasion, microbe want to enter into host cell. Colonization, multiple aganum. Then evasion of host cell. Host cell that want to evade from the host cell. Then damage and disease to tissues. After this multiplication, all cells will infect our host cell. Then after the infection particular time, it automatically exit from the body, because our human system produce some immunity after particular infection date, right? So that is automatically exit. Survive in environment is a very important thing. If the one person get infected with particular microbe, that after some period that person get immunity, so the microbe will automatically exit from particular one human host or any host. In the so the microbe want the host, another host. This period the microbe want to survive in the environment. That may be in water, that may be in air, that may be in soil, any surface. Okay, that need a environment, survival environment. So these are all the order. They are given in question in rearranged format. Please follow. இதில் மாற்றி மாற்றி கேட்டிருந்தாலும் we want to understand the process and give the answer, because we are confused with the steps, because adhesion, transmission, invasion, these are all the confused steps. So please observe these 8 steps for your understanding. Okay, next is pathogenesis. These are all some important points in this particular topic, the host pathogen interaction. Pathogenesis. The pathogenesis means the causative agent produced some toxins. If all microbes are producing some compounds, right? So that produced compound will cause some misleading to our regular pathway. That so only they are explaining, that is pathogenesis. The foodborne intoxication. Intoxication means in that is not regular to our host. The that produced by the microbe, that abnormal compound causing the or misleading our regular pathway. The misleading of this particular this produced so that may cause allergy, that may cause inflammation, so it's called as the toxin. These toxins are the overall term is called as the pathogenesis. Then invading the pathogen to the host surface. After the pathogen, that understanding the particular word, the invading pathogen on host cell surface, because we are asked this question that produced toxins that are damaging the host cell. For they are in this the Vibrio and the Corynebacterium are producing some toxins. They are given the bacterial and toxin name. That is that is also many of the part C questions are match the following questions. Please note, many of the questions are match the following questions in the part C. In that question, they are asked the bacterial name and toxin name. Many of the maybe you have a question, why you need to tell just to give a introduction. You please go with the example. I'm last time I'm prefer that, because I'm telling the example with the questions, but some of them are not understand the basics only. I'm telling this, don't mistake me, and I will send the images. Surely I will send the images. Okay, they are given the bacterial name and toxin name. You just want to find the correct match. This like kind of question is there. Evade the antibody detection. Okay, this because some of the microbes are not recognized by our immune system. So like tuberculosis and plague are not found out by our immune system. The same example as in corona, our immune system will not recognize at the 14th day, before the 14th day, so that cause some infection. Like the same process, the antibody is not detection. Okay, these are all the 5 steps in present in our already I am told, right? Okay, what happen in between the all steps? Transmission. Okay, transmission happen in skin, right? How it's happen? It may cause by cutting of particular portion, that's abrasion, or maybe the puncture. Puncture means it's just a punch, or maybe it caused by the wound. There are many way of transmission is there, right? Transmission route. Okay, sugar patients are having a wound, right? They are not getting cure. Why? There was some microbe present in particular wound. That microbe will get benefited through the wound and through through the wound, but our host will get affected. This is one example. Another one is so this is the example for the wound cutting. Another example is the mucous membrane. Through the mucous membrane, it will enter into the gastrointestinal tract or respiratory tract or the urogenital tract. Gastrointestinal tract, that microbe present, we all know, right? That E. coli, that kind of respiratory tract. If you have a wheezing, many of them may be experienced. We feel some abnormal inflammation in nostril root. That abnormal inflammation caused by the allergic microbes. That microbes will attach, enter into the respiratory system, so our respiratory system will get alert, Okay, someone enter into our respiratory system. Due to this signal passing, our respiratory tract will, or the nozzle tract nodules will get congested, so wheezing will happen in the particular person. These are all the another one. So the transmission route is skin and mucous membrane. The skin caused by some way and mucus. And then another adhesion. Tissue tropism. Tropism is where in this adhesion they are asked the way of adhesion and signaling molecule. Okay. Bacteria cell having this protein and cell surface interact with host, and virus having this protein in cell surface, that is interact in particular host. Another one is and likewise they are asking, given the bacterial cell surface protein name and another side disease name, you want to do the match the following. Okay, in this tissue tropism, specific species specificity and genetic specificity, these are all three are very important thing in adhesion. Okay, what is tissue tropism? Pathogen that prefer the specific tissue. Okay, again I am enter into the corona infection because that spike protein need only ACE2 receptor. Where the ACE2 receptor present in cell surface, the epithelial cells are having the ACE2 receptors. So these are the specific tissue specific interaction. Another one is specific species specificity. Species specificity means it not affect all the host, it only affect the human host. That is the species specificity. Another one is a genetic specificity. Okay, only some diseases are happen in particular genetic species, not affect the humans, it not affect the animals, tigers, it not only affect the elephants, only affect the lions, that kind of is genetic specificity. So they given the disease name, ask the whether it is a species specificity or genetic specificity or tissue tropism. These are all the options. They are given on particular disease name. They ask the what kind of adhesion happened there. Okay, invasion. Next is invasion. Please be wait, because due to the some internet corruption, I am not. I'm lag sometimes, so I am taken another 10 minutes. Please wait for some time. I will finish as soon as possible. Invasion. Microbe being invade into the host and produce. Okay, for that production, that may be, that is two types. One is the bacterium and is a viremia. Bacteremia is a presence of bacteria in particular bloodstream, or viremia is a virus related thing. Okay, now that they will providing the disease name, particular disease name, ask the question, the invasion happened due to the viremia or bacteremia. These are all maybe the question. Fully they are not asking the direct question. Full questions, all questions are indirect only. Colonization, I told right that depends on the host and cell interaction. We having the different we will different species value, species count. Multiplication of pathogen organisms where toxins are produced. These toxins are may help to humans host cell growth or may not. The host being shown the sign is a septicemia. Okay, in a septicemia means blood infection where the bacteria are reproducing. During the bacterial reproduction, that blood in that there is a blood infection or happen. That this blood infection is called as septicemia. Okay, another thing is certain pathogens or less count. some having the larger count. Depends on this microbial count, we will classifying the disease name, and that Shigella is a microbe which is colonized in 1 to 100. That infection is very low infection, but Salmonella cause nearly more than 1 lakh and 10 lakh. That cause heavy infection. The infection rate is depends on count also. Sometime the more count trigger our immune system. So immune system find the alternative to defend this particular disease. So not that depends on the disease and pathogen, we can classifying it into different types. That they may ask, what is the colonization level of Shigella? They are providing the 1 to 10, 10 to 100 for and in the one side they are given the microbial name, another side they are providing the colonization count. You want to match it. We all know bacteria will equally will get colonized in human body. Within 20 minutes it will multiply in many count, and also it will kill by our host cell in particular rate. That also want to we know. Please understand, the bacteria having 10 the Shigella having 10 to 100 count during the colonization, Salmonella having the more than 10 lakh. Okay, then evasion happen in the intracellular in the innate immune system or pathogen. That evasion producing the intracellular pathogen in live inside the host cell. Intracellular pathogen that live inside the host cell, because that intracellular pathogen will maybe affect or maybe the friend to particular host microbe. This based on this intracellular immune system only, we can tell it's a synergic or that host pathogen interaction types, right? Based on this only we can tell the mutualism, commensalism and parasitism. This evasion innate immune system will decide the interaction, host pathogen interaction. Okay, next is I'm Leucodin is I'm going with this example. Membrane damaging toxin. Okay, this toxin kills the phagocytosis. Phagocytosis means that is fully engulfment of this particular bacteria, the host, right? It will stop by the particular leucocidin. We will explain with different example. They are providing the toxin name and also the human. First I am said, right, the bacterial name and toxin name. Another type of question is, are human or host cell produced some toxin against pathogen? So they are providing the disease name and toxin produced in during the particular disease. These are the one example, that leucocidin. Another is it's an for survival of pathogen in human cell or host cell, that microbe need to adapt with particular chemical, or that microbe want to produce the antagonist against the particular chemical. These are the invasion of host defense system. Our host defense system having the different mechanism, depends on the mechanism we will tell. Okay, another is the protease produced in particular microbe that allow the pathogen to avoid the antibodies. That protease will degrade the antibodies, so that so we can't activate the immune system. Another is catalase prevent the digestion of an engulfed pathogen. After the engulfment, that microbe will not digest by the catalase, so the microbe will colonize in particular host cell. After the colonization, it will automatically evade from the human cell or host cell. It's a basic information. Another one is alternation of host cell behavior. What happen in host cell behavior? These are the two examples. One is Salmonella and Shigella. They are causing the ubiquitination process. Ubiquitination, during this ubiquitination process, just there's the usual process produce some substrate, activate by the some substrate, produce some product, right? But this ubiquitination process, that is not silently deactivated and activated by the another enzymes or another molecule present obtained from the Shigella or Salmonella. So in the same case, they are providing the process ubiquitination, catalyzation, proteation, the hydrolyzation, providing the disease microbe name and the process. Okay, in digestion system, that are Lactobacillus doing the hydrolysis of polysaccharide into the molecule. So only if you are taken yogurt means that useful to your digestion system. If you are taken a heavy food, that if you taken a curd, that curd will digest the heavy food. It's a basic principle behind the microbe and microbes producing some proteins or enzymes that will digest our human system. That that hydrolysis. Hydrolysis means the breaking down of polysaccharides. Polysaccharides present in our food, that polysaccharide will digested by the particular microbe present in intestine. This is the process behind this, the alteration of host cell behavior. Another is viral interaction. Viral interaction means it's a normal, once the viral enter into the body, it's attached on the cell surface, release the product in human cell. After that release, the human cell and viral DNA will interact with that manipulate in naturally. So viral gene attached to human gene, the human cell produced to rearrange the produced the microbes, the viral protein compounds. That viral proteins compounds are arranged in particular manner after the particular period that will evade from the human cell. That cause the immune response. Immune response will responsible for our disease. Okay. Then pathogen induced diseases in animals. This is a very important topic. Sorry for the delay, because I must need to tell, because last I'm taken from this one article, but last year December they are asked the question based on this. They are providing the animal name and major infectious disease and species name. Okay. In this Western taught us it caused it having different diseases. This omicity is a one disease which caused by the Saprolinea species. Saprolinea species, due to this infection, these toads are not able to move from particular place to another place. This is not this is spreadable disease. It is very high infectious disease. So they are asking like this, providing the name and providing the infectious disease name, and also providing the infectious species name. A very important one, please listen. Animal name, infectious disease name with infectious microbe name. You want to match all these three things, then only we will get the answer. Please refer this table, and also I am sending on some other tables also. These are some examples. Okay. Next, only one topic, the cell fusion and cell with the normal cell and abnormal cell. This is for normal cell. Normally, if you have a one transformed cell, self-renewed cell, stem cells, both infused both, and it causes some cancer stem cell. Another thing is differentiated cell, stem cell, that different cells are fused with each other, that are producing heterokaryon, then only producing the synkaryon. After that is called as the cancer stem cell. These steps are they are asking. First step, the differentiated cell, stem cell, after that heterokaryon, synkaryon, then only cancer stem cell. So only I put this flow. They are providing in order. You want to they in rearranged order. You want to find the correct order. They are providing the experimental value. During the cell fusion factor, after addition of cell fusion factor, you get only heterokaryon. After chromosomal loss, the lot of due to the chromosomal loss, the heterogeneity will also loss. That loss of chromosome causes the synkaryon. It's a human cell, normal cell. Again I am telling, again I am repeating, the normal cell and the human cell, the normal cell and the human transformed cell interact with the stem, that make a synkaryon, that transform. Another type is differentiated cell, that is transformed differentiated. This is differentiated. This is normal one. Another one is a viral an abnormal interaction. This is normal interaction. This is abnormal interaction. Unwanted thing will interact to host, means that is abnormal. That caused by the cell surface receptors, cell surface proteins. That because that is new to host, right? That is abnormal interaction. Okay. Tomorrow's topic is cell signaling hormones, cell surface receptor. Actually, sorry for this today inconvenience, because due to the network connection I am not able to interact better. These are the tomorrow topics. Tomorrow I hope at 5:30 I will be here. At before 9 o'clock I will send the PPT and the material to your Gmail ID or Google Classroom. Every day you follow this channel, because every day morning, no need to wait. At 5:30, after 5:30, automatically this YouTube link, we will provide the YouTube link, activate the live link. So just enter into the channel and go with live. If you have any questions, please ask in chat. I try to answer. Thank you all. Okay, if you have any feedbacks, please tell in chat, because no problem. If you have any feedback, please tell in chat. this is the first day. If you are tell in this first day, I will rectify the problem. Tomorrow I will do my best. Okay. Tomorrow I'll give some tricks and example questions, sir. And I will. I'm also send the PDF to your mail ID and your Google Classroom with the within a 4 to 3 pages tricks. I'm having on PDF. I'll send. Transformed cell and differentiated cell. Okay, human cell. If you have one human cell, that is divided into two, means that is the differentiated. You have one human cell that is differentiated into two cell, means that is differentiated cell. Transformed cell means one cell that having the another viral particle or viral genome or bacterial genome. They are manually inserted a particular gene in the one cell. That is the transformed cell. Okay, tomorrow I will add some pictures. Sorry. Okay. Tomorrow I will add some examples and pictures. Kavya, ma'am, actually differentiated transformed cell means the one cell having manipulated gene. They are manually inject one particular gene, bacterial gene, viral gene or any human host gene in particular one cell. That is the transformed cell. Differentiated cell means the host cell automatically differentiate themselves. There is no manipulation. That is automatically differentiated. Thank you all. If you have any suggestions, please comment in chat box. Sure, ma'am, I will send the PPT slide. Normal flora. Our eye flora is a normal flora. It's present in our eye in long time, that not affect the eye, but the flora will get benefited. Is there multiple choice evaluation during the completion of topic? If so, it help. Yeah, sure. I will make an arrangement, multiple choice questions evaluation. So, send the pic of class. Send the pic of to classroom. Sure. I will send all the pics to Google Classroom. Sorry, in PPT having all the images. Yes, today I will send the notes, PPT and videos, related videos. Heterokaryon, the name itself we can understand. Hetero, two different. In synchronizing means it's a similar form. So only in first image I told this having transformed cell, this having the synchron, but here differentiated cell, that differentiated cell having different nucleus that are enter into one particular cell, so it's called as heterokaryon, different nucleic genetic materials. Video class recorded I will send to your Google ID and Gmail ID and Google Classroom. Yeah, I will send the Google Classroom ID, and I will send the previous year questions to your Google Classroom. Yesterday is an first day, so I am not able to send that. Today I will send. Okay, example questions I will send. Thank you all. I am going to end this session. If you have any feedback, please tell in chat. Okay, ma'am, I will send some questions. Tomorrow topic is the cell signaling hormones and their receptors. These are fully image oriented. Tomorrow section is fully image oriented and full that is I will explain with the pics. Yeah, tomorrow I will be earlier, and 5:30 I will join with you. Today I had some signal problem. Tomorrow I will cope up that. Thank you so much. I'm going to end this stream.